HIV drugs. Now, HIV drugs, HIV drugs has classes. HIV drugs has classes such as we have the first class, we have a class called the, you must know, you have to go back to our previous videos. You look for the way how the HIV enters the human body CD4 cells. Now, because these drugs, the way they work according to the way they design these drugs according to the way the virus enters the human body. Now, the first class is based on how the virus starts to enter the human body. Class number 1, we have the adherence inhibitor. Adherence inhibitors, these are of two types. We have the adherence inhibitors. We have the two. The one is attachment inhibitor, and another one is fusion inhibitor. One is attachment inhibitor and one is a fusion inhibitor. Here we have, in the attachment inhibitor, we have a type of drug like the for stem server. And here we have another a type here under fusion. We have a drug called we have a enfuvatide. We have enfuvatide. Now here this attachment inhibitor, like the for stem server, we have given us our example. This one, the way it works, it attaches itself on the P120, which comes into contact with the CD4 molecule on the T helper cell. Let's see the example. Now this is our T helper cell. It has a molecule called CD4. These molecules are all over the cell. And the virus which wants to enter this cell, it is here. This cell, this virus, it has the P120. This one here, it's the P120. It is all over its envelope. So this is our HIV virus. This is the HIV virus. It wants to enter into the cell, the T helper cell. So how does it do it to enter this cell? It first attaches itself to the CD4. It first attaches itself to the CD4 molecule. And later it starts its courtship from there, and then later it enters into the CD4 cell. Now, here, our drug here, this attachment inhibitor, it comes and inhibit this attachment here by coating the P120. And once it coats the P120, this virus will fail to attach here. So this is the mode of action of how this drug controls this HIV. Now we have this fusion inhibitor. You know, after this one, after this virus, if you have not used this drug the attachment inhibitor, and we have this whatever, this is our HIV virus, this is our T helper cell. Now, this one, if it has now attached on this one, and we want to control this one, the way this drug works, it will come and coats this thing, which is called the P. This one is called P41. So once it coats this P41, you know very well, in our previous video, we saw how HIV enters the human cell. So in our previous video, we saw that this P41, after seeing that this one has already attached on the CD4 molecule, this one comes and enter into the cell. This one comes and pierces into the cell membrane of the CD4 cell, of the T helper cell. It pierces into the T helper cell membrane here. So once what does this drug do? This drug comes and coats this P41, hence prohibiting this cell from fusing and entering into our T helper cell. That is the mode of action of this first class, of these first two class, the first two subdivisions of the first class. Now, our second class. This is the first class, adherence inhibitor. Our second class, once the cell, we have the reverse transcriptase inhibitor. Now, once the virus has not been controlled here or here, and it manages to enter the cell. Now, if it enters into the cell, the CD4 cell, or the T helper cell, this is our T helper cell. Now, the virus has already now managed to enter the T helper cell. And now, in the T helper cell, what happens inside here? When you check in our previous video, what happens inside here is that this reverse transcriptase, these enzymes that we call the reverse transcriptase, will move along this RNA, and this RNA, and it will move along this RNA, and it turns it into DNA, Now, what this drug will come and work on this and prohibit this from going on. How does it work? Here we have two subdivisions. The first subdivision is the nucleotide or nucleoside reverse transcriptase inhibitors. These drugs, here we have examples of this drug. We have like zidovudine, which they call AZT. Then we have the lamivudine, which we call the 3TC. We have tenofovir, we have these drugs. We have tenofovir under this nucleotide cells inhibitors. We have the abacavir. have abacava. We have also the D40. This class of drugs, this one is under the is example of this is the example of some of the drugs we use in the nucleotide reverse transcriptase inhibitors. The mode of action of these ones they do work like mimicking. They mimic like they are the like they mimic like they are the building blocks, like the natural building blocks of the DNA, hence preventing this reverse transcriptase from moving along with this one to change this one into the DNA. Hence the process is stopped. So that is how these ones work. These ones. Then under this class, the reverse transcriptase inhibitors, we have the one which is the nucleotide. I mean, actually, we have looked at the nucleotide. We have the non-nucleotide reverse transcriptase inhibitors. Here we have drugs like efavirenz. Here we have drugs like the nevirapine. We have entrevarin. Now these ones are example of these non-nucleotide or nucleoside reverse transcriptase inhibitors. These ones, their mode of action, these ones they just come and bind. This is the T helper cell. This is the T helper cell. Now, for the enzyme is here. This is the reverse transcriptase enzyme. Instead of it has to move along this one, along the RNA to form the DNA. Now, this one, instead of moving, when it prohibits it, it comes and bind to this reverse transcriptase inhibitor. It binds on this reverse transcriptase enzyme and stops the process of it to move around and change the RNA to DNA. Hence, this process is stopped. So the third class, we have what we call the integrase inhibitors. We have another class. We have another class of the antiretroviral drug called integrase inhibitors. Integrase inhibitors. Now here we have drugs like the bictegravir. We have drugs like bictegravir, raltegravir. We have dolutegravir. Like the elvitegravir. So these ones, as you check in our first video, we said that the drugs, we said that the virus, when it is entering into the body, this is our CD4 cell. Now it has already bypassed the first stage here. It passed, if the virus passed this stage, it passes also this stage of the new of this reverse transcriptase. It reaches this stage. Now, this stage we want to eliminate it at this stage. What do we do? Here it has finished to reverse transcriptase. Now the DNA enters into the nucleus of the host cell. Now the nucleus of the host cell, once it enters here, the DNA formed by this virus moving along the RNA. This virus eventually moves from the cytoplasm and enters the nucleus. And once now it is in the nucleus, how does this drug work when the DNA has entered into the nucleus? Now, the nucleus, once the DNA is in the nucleus, it finds there the DNA of the human person. Now, if it finds the DNA of the person, it brings its DNA, the virus brings its DNA and fixes it together and joins it to, because the integrase enzyme joins the DNA of the virus to the DNA of the human being. Now, this drug prohibits this from happening. So it binds to this integrase, to the integrase enzyme, and prohibits it from fixing the virus DNA into the host cell DNA. So that's the mode of action of this drug. This type of drug, it is powerful, by the way. It is powerful. It is powerful, this type of drug. And its side effect is not all that too bad. Although some people said that they get some insomnia, some people may fail to sleep, but with the time they get used. So this type of drug, it is recommended, it is highly recommended even these days, to be included in the combination of the heart drugs. Actually, they are heart drugs. So now, it is so powerful. That's what I wanted to tell you about this drug. Now we have the fourth class of drug, which is the protease inhibitor. This protease inhibitor, here we have drugs like atazanava, lopinava, ritonava, ropinava, rotinava. We have also darutinava. We have darunava. Have also darunava. So these ones, if the virus has bypassed this stage, and it has successfully fixed its DNA into the human DNA, and now this human DNA starts producing RNA. The RNA, this messenger RNA, this is our T helper cell. This is our T helper cell. Now this is the nucleus. Now it has successfully fixed in the what? its DNA into the DNA of the human person. Now, it starts producing the RNA, and this messenger RNA, they come back to the cytoplasm of this T helper cell. Now, it is this messenger RNA that are used to synthesize the proteins that help to rebuild back another virus, which will infect another cell until they infect another person. So, these ones, they use this DNA, this protease will be here in the cytoplasm, and it is this protease to come and cut these messenger RNA and cut them into small substances that will help the virus to grow and become another virus. So what these drugs do, they come and bind to this protease enzyme and prohibit it from doing its work of cutting, of breaking these RNA into smaller pieces to build another virus. So that is it about the drugs of HIV, their classes, their mode of action. Don't forget to subscribe, because when we put another video, you will be the first person to receive the notifications. We have videos in 2 languages. We have videos in Lusoga, we have videos in English, because I was asked to do most of these videos in English. But initially, I had somebody had told me to do it in Lusoga, so I have 2 requests. So I do videos in Lusoga and another video in English. There is another video also in Lusoga. So you choose what you want to get. You just subscribe to our YouTube channel, which is Dr. PCM. Just subscribe to this YouTube channel, Dr. PCM, and get to know more about what we teach. Thank you.